Okay, I think we will get started now. Good morning, everyone. My name is Gabriela Segal, and I am an application specialist at the Biological Optical Microscopy Platform here at the University of Melbourne. And today we will be continuing from our lecture tomorrow, and we'll be covering sample preparation, experimental applications, and a bit of image ethics. First of all, we would like to acknowledge the traditional owners of the land where we work and study. So you can read this here. And a little bit of what we'll be covering today is we'll go into advice on sample preparation. Then we will move into some of the applications, and mostly applications that are available within our platform. And then we will talk a little bit about ethics and what it's okay to do and what is not okay to do in terms of imaging and image analysis. So sample preparation. This is one of the key things from your microscopy experiments. And usually this will determine a lot of how successful your microscopy experiment goes. We will give you a few of the recommendations especially for using our instruments or imaging in some of our microscopes. And one of the first things is all of the best imaging is done near the cover glass. So the closer you can have your sample to the cover glass, the better image quality you will obtain from this sample. So if you can mount for example, your cells on the cover glass instead of the glass slide, that will result in better imaging. The second one is actually quite key. The cover glasses that most of our microscopes need are 170 micron thick or a 1.5 cover glass. And the lenses that the microscope have are adjusted for this thickness of cover glass. So by having this size, your imaging will also be better. You can see here in the bottom a little bit of an example of what would happen if your cover glass is thicker, and how, for example, in this case, it's just a bright field image, and how the image quality really decreases when the cover glass gets thicker. These 1.5 cover glasses are important for adjusting to the right point. And especially if you are doing something like super resolution, they need to be a very precise size, which are the 1.58. Next thing, you heard a lot about yesterday from fluorophores and how you need to select the best ones for your experiment. But not only that, you also need to match the microscopes that are available to you. So if you have a certain laser that can excite your fluorophore, then that would be a good choice, for example. And the next thing would be to titrate concentrations. So if you're doing transfections, to adjust the amount of DNA you use, the fixatives you use, the antibody concentrations, and really, really important, positive and negative controls. So your microscopy experiments, even results in images, people tend to forget that they are also scientific data. So you do need positive and negative controls that shows that everything is working as it should. So you want to use some gentle fixation that will preserve your sample and image your samples as closely to your preparation as possible. For mounting your samples, that's also quite important. you want to have a mounting media that matches the refractive index of the lens you're using. So for example, in here, we can see what happens when you use a mounted that has 1.51 refractive index versus a 1.33 refractive index, and how you can see the image quality is much better in here because we are matching the refractive index of the actual lens, which is an oil lens in this case. Also, really important to avoid mountains weakly, as they would also result in more background in your sample. In terms of general sample preparation rules, you have sort of this decision tree a little bit in here, where if you are imaging cells or tissue, you have two options: either go with a live sample or with a fixed sample. When you're imaging in a live sample, you're pretty much left with two options: either fluorescent proteins like GFP, like something you heard yesterday, or dyes, something like DAPI that intercalates in DNA. When you go into image fixed samples, you have those two same options, but now you have the extra option of antibodies that can recognize specific antigens within a cell. If you are going to be doing live cell imaging, you very importantly need to keep your cells alive. And we do that by controlling temperature, controlling the gas environment, the humidity that the samples experience. So most of the incubators, we have some humidifier. And if you want to reduce evaporation as well, we can use gas permeable seals. The cells also would need to have media, like food, to be happy. And then in terms of which microscope you would use, it will depend a bit of how dynamic the process is, which one you would choose. We'll cover a little bit about some of these microscopes today. You heard a lot about confocal yesterday. When choosing the right type of microscope, for example, we have something like the spinning disc that is quite a gentle and fast imaging technique. That would be really good for quick dynamic process. Then we have confocals that have a resonance scanner that can scan quite quickly the samples. Other options like the Eriscan Fast or Eriscan 2 that can scan several lines at the same time that are more gentle than the regular point scanning confocal. And other things like lattice light sheet, which we do not have available in our platform, but would be quite gentle too. You want to select a fluorophore that should be as bright as possible, so you don't need too much light to excite it. And you want to use fluorophores that require long excitation wavelengths. So, for example, avoiding UV. You want to reduce the phototoxicity, and again, UV is not really good for the cells, so you want to use higher wavelengths for this type of experiment, and with low excitation levels, so your cells continue to be happy through your experiment. How you mount your samples, it will depend highly on what type of samples you have, but there's a lot of options. One of the chambers that can be used in a lot of the microscopes is the Eisner chamber, which is basically composed from two cover glasses that sit on either side of these metal rings, and it's sealed. Other options are the fluoro dishes or latex or EBD. There's a lot of vendors for this that are cover glass bottom thickness, and they are specialized for different applications as well, depending on what type of samples you have, cells, organoids. There's a lot of dishes that might be able to suit your applications. Very important, how you fix the sample could really determine what sort of results you get. in terms of signal, in terms of structures. The most common fixatives, we have glutaraldehyde, which is very strong. It keeps the morphology really well, but the staining is not so good. Formaldehyde, which is one of the mostly used for fluorescence. It's weak fixation, keeps the morphology a little bit poor, but it's good for stains. Acetanol methanol could cause too many artifacts, and also we have some of the newer ones, which is glyoxal, something that is sort of in between the glutaraldehyde and PFA. And there's a really good paper here where they compare lots of different stains, how do those structures show when you fix them with PFA or formaldehyde versus glyoxal. So if you're gonna start imaging something, it's always a good idea to find what people are using for fixative for that particular structure. As you can see, some of the structure look much better with glyoxal and PFA. Some of them might look relatively similar, but the decision will be based on the stains or the structures you're trying to see. If you are imaging now fixed cells, in this case, you would grow or attach your cells to the cover glass, and we use that by using some positively charged polyl-lysine or cell tag that allows the cells to stick if they are non-adherent, or if the cells are adherent, they will just stick to the glass. Then we go and fix with either of your fixatives that we spoke before. And important, you need to titrate the concentrations and conditions because those will affect how your structures will look. Then we're going to permeabilize the cells. You can use detergents like Triton or Twin 20. And depending on whether you need to penetrate, how much detergent you need, and how long you need to permeabilize the cells as well. So different agents are different, work different for different areas of your cells. Next thing, quite important, is you want to block any non-specific binding, and you do that by either using bovine albumin serum or by using the serum from the secondary antibody species. This will reduce any sort of non-specific binding of your antibodies. Then we go with antibody labeling and mounting the sample. If you would be doing some paraffin preparation, the process is slightly different. So in this case, you isolate your tissue and you use some chemicals fixation like formaldehyde. You embed your samples in paraffin and dehydrate them. Then the samples are sectioned in this little machine we see here, which is a microtome. You rehydrate your samples, and then you need to recover the antigens by either doing heat or enzymatic recovery. Then you block an antibody label. If you do tissue sections that have been cryopreserved, the first part of the cryopreservation is you will do, again, chemical fixation. Then a sucrose infiltration for the cryoprotection of your sample. You will put your sample in OCT. That will allow to freeze homogeneously the sample. And then the samples can be cut in a cryostat, which is this little machine here. You cut thin sections, and then those sections are they let dry, and you store them. You can store them for a longer term in -80. If you're gonna straight away do the stainings, you can do rehydration, block, and antibody label afterwards. And then we have the option to do antibody labeling. If you are using antibody labeling, you need to be very aware of the species that you're going to be using if you're using a primary and secondary antibody. So you first incubate with your primary antibody, you wash through, then you incubate with your secondary antibody in this case. Like in the example here, for example, if we have a GFP antigen, we use a mouse anti-GFP for primary antibody, and then a secondary, which is a goat anti-mouse. Then we wash those samples, fix the samples, and wash again. That's optional. And then we mount the samples. And you can see how in this case, when you're using a primary and secondary antibody, you are a little bit further away from your antigen, but it can also result in some amplification of the signal as well. And very important, it is really important that you, at the very end, mount your sample by using a mounting media and putting it, depending on what sort of format you're going to be imaging. At this point, just to check if you guys have been paying attention, we will have a little quiz. And so you should be able to see this now. So there are 2 questions in here. Oh, sorry, that's not the that's the wrong one. Now you should be able to answer this one. There are 2 questions in here. So feel free to answer. We'll give a few minutes so you guys can answer this. Most of the things I would have mentioned during this past couple of slides. Just last 10 seconds. I think most people have answered the questions already. Good. So if I end this and share the results so we can, you guys can see. What is the best choice of cover glass for most conformal microscope application? In this case, as I mentioned before, in most cases, we are looking at a 1.5 cover glass. So it's 170 micron thickness, which most of you got right, which means not everyone was sleeping. And then what should you take care when choosing an antibody panel? And in this one, the correct answer is the primary. Oh, that's actually incorrect. The secondary should target the species of the primary antibody, which is this one here. So 76% got it right. I just wrote the wrong one in there. So we can keep going. If you have some more questions, we will address them at the end. So just feel free to put them in the chat, and then we will do our best to answer as much as we can. So we'll move on to the next part. So feel free to put your questions in the Q&A, and then we will answer them at the very end. Yeah. So the next thing we're going to cover here is the experimental applications. In this case, we're going to be covering what are the types of microscope and techniques you can do. And one of the things is you can do microscopy across a lot of the scale sizes, but the type of technique you pick for one or the other depends on so what you need from the imaging. So we can image really large things up to full tissue, full organism with fluorescence microscopy. And we can move into have more into details of the tissues up to 100 microns, things like that. Or if you want to go more like cellular resolution. If you need to go beyond what is this red line here, which is the diffraction limit, you need to delve into the super resolution techniques. And if you need to go into more like really Armstrong resolution, we are looking into electron microscopy, which will not be the topic of this lecture today. So first of all, we're going to talk about the techniques that allows us to get the cellular resolution. And we're going to cover light sheet microscopy, multiphoton microscopy, slide scanner, high content imaging, and wide field microscopy. So light sheet is the first technique, which allows you to image whole organisms. In this case, we can see this is actually a human embryo that have been imaged, and you can see the sort of details you get from this type of imaging. So light sheet microscopy works in a slightly different way than what you would be used to. In light sheet microscopy, what you're doing is you're illuminating your sample using a sheet of light that can be generated by using cylindrical lenses or objective lenses. And that means that you are only really imaging a thin section of your sample at a time. And you're detecting perpendicular to your illumination. And you are able to then reconstruct all the volume information from your tissue or from your organism. So in this case, we are able to then obtain volumetric imaging from, in this case, was Drosophila embryo, zebrafish embryo, or things like that. It allows you to get up to a cellular resolution, and the samples are imaged using a dipping lens. So in this case, the refractive index is actually quite important for matching sort of the refractive index of your imaging solution and the refractive index of your sample. Samples needs to be optically clear, and we're going to speak a little bit about the optically clearing samples. And in some cases, samples are intrinsically clear, which are easier to image, like some of these embryos. So for tissue clearing was developed in the early 1900s by Spattenholz. And so it's not a very new method, but it only got traction once some of the microscopes were available for imaging clear samples. So what it does, it removes some of the components from the sample, either by putting more water into the sample, by removing lipids. And that makes the sample homogeneously transparent. So you can see, for example, here from brain, after one of the process of clearing, you can see how you could read all through the text that is behind it. And the types of methods for clearing are basically, there's a lot of methods, but we can separate them in two main groups. One is using organic solvents. They remove the water from the sample. Then they match the refractive index with an organic solvent. It is not always applicable for the fluorescent proteins. And in these methods, we have things like BAP, disco, 3-disco, iDisco. There's a lot of options in there, and ethyl cinnamon. It is quite a quick method, but it also results in some shrinking of the samples. So that's something to be aware. On the other hand, we have the aqueous phase. In these methods, we have things that first remove the lipids from the samples, and then we match the refractive index. And in some of these cases, we are introducing water into the sample by passively adding solutions, or we can use things like electric current to clear the samples away. And in these families, you have clarity, cubic, CDB, switch, and also, again, a lot of options up there. And not every clearing method is good for every tissue. So if you are thinking on doing any sort of clearing, it is first a good idea to have a look on the literature, what people have done. So then it will be easier to clear based on a protocol. In our case, we can image in our system in either of these solvents, so solvent or aqueous phase. And one of the applications is here. So this is guinea pig cochlea that has been implanted. And you can see how you could visualize all the hair cells through the cochlea. And this gives you a really nice structure. So in the past, these researchers needed to do sectioning of this sample to get information. Now they can get a full reconstruction and get a better idea of what's happening in terms of tissue response to the implant and what is happening to the hair cells after that. Next up is multiphoton. So multiphoton microscopy is mostly used for imaging live animals, but it's not restricted to just that. It uses infrared wavelengths, so there are like higher wavelengths, 700 to 1,000, even higher now on, that can penetrate the sample deeper. So what the two-photon principle is that instead of just using a single photon, you can excite with two photons that have a higher wavelength, and then you can achieve the same excitation of your fluorophore and the generation of fluorescence. So you can see here some examples of fluorophores that are typically used, and how you could use a single photon like you have in the confocal, but when you use the two-photon excitation, you're almost doubling the wavelength. And in most cases, in multiphoton, we can penetrate quite deep into the tissue. So we are having users imaging into brain, lymph nodes, liver, deeper into tissues that otherwise would be impossible to penetrate with other techniques. And an example we have here is from Alice Abe and Scott Muller, where we can have, in this case, is the mouse skin that hasn't been infected with herpes, and herpes can be seen in purple there. We can see the collagen, which is just the blue structure around there. And we can see two types of T cells. So we have the ones that are non-specific for herpes, which are the red ones in this case, and the green ones, which are the ones that are specific for herpes. And you can see how they have different behaviors. So you could then analyze what is the speed of these T cells? Are they always sort of surrounding the same areas, sticking to an area, or are they moving quicker? So you can do a lot of analysis in the tissue itself without having to really fix CAD. So it gives you a really dynamic view. Next up is our slide scanner. So slide scanner allows you to image in very high throughput fixed samples. So tissue sections or cells in slides. So slides with cover glasses, very important. They need to have cover glasses for this system. And it's based on bright field or fluorescence. It has a camera. The one we have available within BOM is the AxioScan 7, so you can load up to 100 slides. And we have all these types of lenses, all airs of 5, 10, 20, and 40. And it gives you a really high throughput output, so you can image really large pieces of tissue and just load 100 slides and get all the images through. An example is here. You can see how we have the slide here that has the pieces of tissue outlined. You can first get an idea of where every tissue is. You could zoom in into one of the particular ones that you're interested in, and then get more details of those structures and move on into higher magnification, for example. So it's a really good instrument for screening. And if you are dealing with thin sections, it is a very quick technique. Next up is high content imaging. So high content is for when you, instead of wanting to image just a single dish, you want to image, for example, a 96 well plate. In this case, you are looking to do some automatic imaging, so the system can visit the different wells in your 96 well plate and allows you to do sort of an automatic imaging. You're not really picking wells, you're imaging lots of different areas. So it's unbiased, it can be high throughput, and it's really good for validation or screening if you're testing if a drug is killing your cells, for example. It does require some optimization, and you need mostly on the sample preparation side, and the size of data you generate from the system is also massive. It's not really for specific region of interest imaging or for super resolution, but mostly to get a big idea of what's happening to a larger population of cells. And the applications of high throughput screening are really endless, let's say. So you can do basic things like cell counting, nuclear counting, determine lives to death. And I'm not going to read this whole list, but you get a lot of ideas of things you can do. Some of the most popular assays are things like migration assay, where you have a scratch or a mole, and then you evaluate how many cells migrate into an area after a certain amount of time. You can also do cell tracking. So the system is equipped with incubation, so you can keep the cells alive and happy for the duration of the experiment. You could also analyze morphology. So it is really powerful for doing a very high throughput analysis. Next up is wide field. You guys probably heard a little bit about wide field on your lecture yesterday. I'm not going to go into too much detail on the hardware itself, but wide field, it's an imaging technique where you are really detecting all the light from the different focal planes. And one of the things we can do for wide field images is to then process in a process that is called deconvolution that can result in a higher resolution image. And what is deconvolution? I'm just sort of going to dip into it very briefly. but you'll probably hear a little bit more tomorrow. So what image deconvolution is if you imagine you have a spot of light. Once the spot of light passes through your optics, through your microscope, the spot of light actually becomes more diffuse. So then what you can do is a mathematical process to reconstruct what is the actual sort of ground truth and restore the image. So you can, by doing this, you can increase the resolution in X, Y, and Z. You can increase your contrast and remove noise as well. And it could be quite powerful for some of the imaging techniques. It does require your sampling to be Nyquist or better. So you heard a bit about the sampling rates yesterday. And it works better in a set stack of data because it's gonna reassign the light to the different focal planes. So the more set information it has will be better. And BOM has 2 professional licenses that can be applied to wide field, which is one of the applications, but it can also be used on confocal, on Eriscan, on Lightsheet. So it can be a very powerful tool on the top of your imaging that can lead you to higher resolution, even with using a wide field system to start with. So next, we're moving up a bit into some smaller scale. So we are talking now more cellular resolution. So in this family, we have turf microscopy, confocal microscopy, F experiment, the confocal super resolution will come next, structure illumination, and single molecule localization. So first of all, turf microscopy. This technique is based on the fact that when you have a source of light and you reach a critical angle, all the light reflects back from when it travels from one refractive index to a different refractive index. So in most cases, we have a propagating wave, so we are illuminating everything. While in turf, what we are doing is we are illuminating with an angle. So we are only really illuminating what is called the evanescent field, which is the first 100 nanometers of your tissue. And that allows you to get very high resolution image of those structure. And you can see when you compare, for example, an epifluorescence versus total internal reflection microscopy, you can see a very low noise image and allows you for detecting like a very thin section of your sample. But it does need to be something that it's sort of closer to the surface of the cover glass because you are really just imaging this first bit. You can have an example here of turf, and you can see how you can see the structures, in this case in the membrane. And within BOMB, we have turf available in the Niken spinning disk in Bioscience 2, and also in the Lyra 880, which is located in Bio21. So this type of technique is really good for calcium imaging or cytoskeleton, and also for some of the super resolution techniques that we're going to cover. Next up is confocal. And I think you guys had quite a good introduction to confocal yesterday. So just a little tiny reminder about confocal. So in here, what we are doing is we are scanning our laser to the sample. And then when this signal comes back through different mirrors, you have this pinhole, which is what allows you to have that extra set resolution in confocal. It is a very popular technique, and we do have lots of confocals within BOM. Allows you for having this 3D data acquisition, and you can also have multi-position visiting, so you can do tile scanning if you want to have a full tissue image at the same time. It also allows you for doing some live imaging, depending on the microscope you have. how fast. So if it's the best instrument, some of the confocals could be quite slow because you're scanning point by point, but you have in some of our confocals also some of the area scan fast that can help with this dynamic imaging. You could run some of the F experiments we were going to cover in a little bit. You can cover photoconversion. And in some of them, we have specialized detectors for super resolution microscopy, like the area scan detector, and also spectral detection. So one of the applications you could do is, for example, in whole mounts of a retina, you could image the whole retina, maybe with a lower resolution, and then go and into more detail with a single field of view and image that one at a higher resolution by using the tile scanning or the stage functions. Another one of the applications is spectral imaging. So in spectral imaging, what you could do is instead of imaging a single band pass like you do in most of the cases, you can image all through the different wavelengths and then determine what's the signal coming from different types of fluorophores. So you can either have a multi-channel photomultiplier. So this is an array that covers 32 channels or 34 channels in some cases that can go from 400 to, let's say, 700 nanometers in this case, 750. And you can get images all through the different wavelengths. What results from this imaging is you get the spectral information, so continuous spectral information. And by having a library of the different fluorophores, you are able to then distinguish what's coming from a different fluorophore. And you go through a process that is called unmixing. So you get the signal from all the fluorophores together, and then you unmix what comes from a different fluorophore. And you can determine by having a library of single fluorophore, you can determine which part of the stain is coming from what. And a user here from microbiology, Daryl, was imaging, in this case, up to 9 channels, targeting for different immune cells in spleen, I think. So you can see how, like, when everything has been established well, that you can determine what's coming from one and what's coming from the other. This is also possible to do in other systems that do not have the multi-channel array, but you can image through different wavelengths, which is called lambda mode in some cases, or continuous band mode, depends on the vendor you're using, which would be a little bit slower, but it will give you at the end the same result. So that's sort of spectral information. Spinning disk confocal. In spinning disk confocal, unlike in the confocal point scanning confocal, in here you have a disk and you are illuminating. To show you, so let's go back to that. That's supposed to be an animation, but that's so. Forget about it. That's supposed to be an animation, but it's not working. So in this case, you have a disk, and then it's a bit annoying. It is faster acquisition because you have the disk constantly running, and you have a camera detecting the light. So you can see here in the laser scanning confocal, you're sort of scanning one side, so coming around, While here you have this that rotates and have lots of pinholes that allows you to capture processes that are quite quick. It is using a camera, so that makes it a lot quicker than the point scanning confocal. But it is not applicable for a thicker sample because you get some of the out of focus light. An example is here where you can see, in this case, this outgoing mitosis process that can be quite hard to see if the instrument is too harsh. The cells are really not happy to undergo the normal processes. So you can see how you can see some of these dynamic processes. It is less phototoxic. So it's a really good tool for live imaging. Now moving on about the protein diffusion and interaction methods. So in this case, we have the what is called the F experiments. The fluorescence recovery after photobleaching, which is FRAP, fluorescence loss in photobleaching, FLIT, fluorescence fluctuation, spectroscopy, FFS, and fluorescence resonance energy transfer. So we start by going through what is FRAP, which is the recovery after photobleaching. So in this case, what you have is you have a cell, for example, you bleach an area by putting the laser very, very high. So you're bleaching all the fluorophores in a certain area. And then you analyze the signal and how that recovers after the photobleaching. So then you can analyze the intensity. So you have here your bleaching event. And then you can see how much of the fluorescence recovers. And you can determine from that how much of this protein, for example, is mobile. So the immobile fraction is the part that would not recover, and the mobile fraction, the part that recovers afterwards. So you can use this to study membrane and membrane dynamics. So you can analyze two types of proteins and then determine how long does it take for recovery, or how long, what's the diffusion coefficient of those proteins. You can also study things like a nuclear pore complex, where you can do the bleaching in the nuclear pore and then analyze how the fluorescence recovers after that. Next up is fluorescence loss in photobleaching. So this is very similar to FRAP. The difference is that in here, instead of having a single bleaching event, we have bleaching happening continuously. And then we are analyzing within a different ROI, for example, in this case in the membrane, what's happening to the fluorescence in that area. So by analyzing this type of experiments, what you get is information about the continuity of the membranes and the organelles, so how connected they are one to another. Then we have a family of techniques that is called fluorescence fluctuation spectroscopy that is used to investigate the mobility of molecules. This can provide a lot of information about the concentration, how the molecules diffuse, the number of molecules, and what is the oligomeric state of the molecules. You can either image a confocal volume and then analyze the signal in a fixed spot. Then you can analyze how is the fluctuation in that spot of analyze something like FCS, where you can have a correlation of things that are moving faster or slower. You have other ways of analyzing with these methods, like by using just raster scanning, and there you have things like number and brightness that allows you to determine if something is monomeric or oligomeric. These are highly specialized techniques, so I'm not going to go too much into details into these ones, but we can help you out if you want to do some of this imaging. Next up, which you heard a little bit, is fluorescence resonance energy transfer. So in this case, what we are using is two fluorophores that are quite spectrally close. So in this case, the example is using a CFP protein and a YFP protein. And because these two proteins are spectrally quite close, when they get into close proximity, what it happens is the transfer of energy. So for example, if you have a protein with a sensory domain, and then you have a substrate, and each of them has the fluorescent protein attached to it, when you have the CFP alone, and you excite with the right laser, what you'll get is something that emits mostly in this case at 470 nanometers. But if now the two proteins are together, and then the two fluorescent proteins are in close proximity within 10 nanometers, what you get now is more emission on the 528, which means there was transfer of energy. This technique is really powerful to determine if two proteins are really interacting with each other. Very important is that you need to match the donor and acceptor. So it's basically based on the fact that these two fluorescent proteins are very overlapping, so that you can excite the YFP by using the energy from CFP in this case. And there's a lot of FRET pairs that have been described. So CFP and YFP is just one of them. And FRET applications, what you can use it for, as we mentioned, protein-protein interactions. So FRET will occur in this case if you have a blue fluorescent protein, a green fluorescent protein. In this case, if the two proteins are together, then you get emission at a higher wavelength in this case. Things like calcium sensing. So if a protein gets modified after calcium is by its binding, then you'll get FRET between those two. It's like kinase activity when the phosphorylation changes the conformation of the protein as well, or things like protease where the cleavage of a certain area will, in this case, result in no FRET occurring anymore. For analyzing FRET, there's quite a few ways of doing this. And you can see this acceptor photobleaching, so that's only for fixed samples. So for example, you have your sample here, you do the bleaching, and then you analyze the signal that is resulting afterwards. So that's only for fixed samples. Then radiometric, so if you have FRET pairs, you can determine the ratio between fluorescence before and after bleaching. Sensitized emission, that requires quite a few more controls, but it's a lot cleaner. And then probably the better way of doing the FRET analysis would be by relying on something that is not intensity-based, which is the lifetime. So that makes us move on to what is fluorescence lifetime imaging, so FLIM. As you would have known from your lecture yesterday, in fluorophores have lots of characteristics, and one of them is how long do they stay in the active state before they go back into dark state. So this technique uses this principle where you use a pulse laser. So you send your laser through, and then you determine when is a photon being emitted. So you can measure the time between that. If you measure the time and then you put it within a function, you can determine the lifetime of individual fluorophores. And you can see here, without doing any stain, just by imaging plant section that has different components, how different areas have different lifetimes, and you can distinguish different compartments. For example, you can see how fluorescent proteins or fluorophores are quite spectrally similar. When you do analyze their lifetime, it's very different. So that's very useful for determining, so separating one from another, like multispectral analysis, but also for establishing if two proteins are interacting when the lifetime changes. One example here, these researchers from Melbourne Uni were looking at histone, how compact were the histones, and they had this Fred sensor. So when there's no Fred, you'll get sort of only signal from one of the fluorophores, low Fred a little bit, and then high Fred, you get also emission on the red fluorophore. And if you analyze this, what is called a phasor plot, which is in the Fourier space, just sort of a vector of the lifetime, you can see the changes from the lifetime when you have a single fluorophore and into one where the two fluorophores are interacting, and how that gives you an indication that these two proteins are close together, or in this case, that the chromatin is very compact. Next, we're going to move up into the next set of techniques, which is called super resolution microscopy. So this is a family of techniques that allows you to go beyond the diffraction limit. So as you would have known from your confocal lecture, your point scanning confocal allows you to go up to 250 nanometers, probably the best case scenario. If you want to bypass that, you need to use different types of techniques to overcome this limit. So we're going to speak a little bit about the Eriscan, which is one of the types of detectors that uses sort of like smarter optics to do that. There's other techniques like STED, so immediate depletion, that allows you to get up to maybe 130 nanometers, but we don't have any STED either, STED or 3 times STED within our platform. Structural illumination, which can allow you to go up to maybe 60 nanometers, and the one that allows you to get sort of the highest resolution, which is the single molecule localization that we are going to talk a bit about. So you can see sort of different options within super resolution, and it will depend on what you need to see, which ones you will be picking. But we are talking about subcellular compartments. So in this case, you guys heard a lot about the confocal, which is basically using a pinhole to get rid of any out of focus light. And most cases in the confocal, we're having our pinhole around 1 area unit. What we can do in some of the systems that have the Eriscan detector is we have this 32 array of detectors, and each of them has a 0.2 area unit. And by having this small detector array, what we can achieve is resolve smaller things, so up to 1.7 increase in resolution. And because we have the array, we're not really losing the light. Like if you would do the same in your regular confocal, you will be excluding 95% of the light in most cases. So by doing this, you're still capturing all the light, but you're getting more resolution from your sample. And you can see here how both the axial and lateral resolution increase with the Eriscan. An example is here. So if you have your regular confocal image, and then you put the same image on the Eriscan detector, you get this sort of imaging. Very important, and I would say the most positive thing is you don't really need to prepare your sample in any particular way to be able to use the Eriscan. If you have already your slides ready for confocal, they can just be imaged on the Eriscan mode. And we do have Eriscan detectors in a lot of our confocals located through the campus. Another example here where they use Eriscan detector, but on the top of that, after doing the imaging, the images were deconvolved using Huygens Eriscan deconvolution. And then you can see we can reach up resolution up to 190 nanometer resolution. This is this image on the bottom. So by using this detector, which already increases your resolution, and doing some of the image deconvolution on there, you can increase your resolution even further. So it's something to look up if you are looking into sort of going beyond and you already have samples that could be confocal ready, so Eriscan ready. Next up is 3D structure illumination. This uses illumination pattern that can be sort of rotated in different directions. And with each of the rotations, you get sort of the resolution of so higher resolution image of different angles. Then all those images can be reconstructed into what is called a sim image. And you can see how by having all this exposure and information from the different angles, what you end up having is quite a high resolution image when you compare it to just the wide field system. Next up is the single molecule localization microscopy. So this family of methods bypass really the diffraction limit with a different method. So in this case, you're not really relying on all the fluorophores to be on at the same time, but you're really more relying on the fluorophores, only one of them to be on at a single time. And then you are detecting really single particles, and you can reconstruct later on an image. And a really good example of how the single molecule localization works is this example from the Eiffel Tower, where you have the sort of tower blinking. If you would have different, I'm going to see if we can put it again. But if you have sort of just a few molecules blinking at a single time, then you can, at the end, reconstruct the full tower. And in this group of techniques, you can either use PARN, which is using specialized fluorescent proteins that can be photoactivatable. You can have STORM, which is using, in this case, antibodies and chemistry to get the fluorophores from a excited state to dark state, so to obtain this sort of blinking from the molecules. Or you can use DNA paint that uses oligos and binding of the oligos to generate this sort of blinking. So you're no longer looking at an sort of like an image of a cell, but it's more like the reconstruction of the molecules that were imaged during that acquisition. One example here is from Oliver's imaging, where he was imaging malaria parasite. And you can see how in wide field you could barely see the structures, while after you process, so sort of image the samples and then reconstruct, you can get really distinct structures within this parasite that were like donuts or shoehorse shape. So really pushes the resolution up to 20 nanometers. So with that, we've been covering sort of from the higher scale, bigger things up to quite smaller things, to maybe 20 nanometers. And then if you need more information, so smaller information, then you're looking at some of the electron microscopy techniques. We have another poll to see if you guys are still awake. Let's go back to this one. Okay. So I'll give you a few minutes to have a go at this. So you should be seeing the questions in your screen. Yeah. So hopefully, you'll be able to answer some of these questions about the experimental application. And again, if you have any questions, feel free to put them in the Q&A, and then we'll cover them at the end of the lecture. Yeah. I'm gonna share that up. So maybe this one was a bit tricky. So if you would like to identify subcellular structures, so less than 250 nanometers, the technique that you'll be looking at is STORM. HALE is not actually a technique. I just made that up. Confocal can reach up to 250 nanometers. Then, which technique is often used for full organism, intact tissues? So in this case, we are looking at a light sheet, which can really image big chunks of tissue. Okay. So we're gonna move on to the next and last bit of this lecture, which is a little bit about ethics in imaging. So I'm going to talk a little bit about what things to do and what not to do. First thing up is you should always be saving your files as a raw file extension. So whatever vendor you're using, you should be saving, for example, if it's a size system, a CSI, or like a LIF file. Do not save TIFF or JPEGs, as some of those conversions don't preserve, so compress the files, and also don't preserve some of the metadata. So all of these raw files from the vendors not only have the images, but also how the image was acquired. And you can see how raw file can also help you to then go back and remake a same experiment you made like 3 years ago or something, or a student, or reproduce something that a student did previously. And they can be used in most image analysis software without any conversion. They can be opened in Fiji nowadays very easily. As I was saying, you should keep your raw file, and then if you need to do exports, you should do TIFF, but not compress. So if you are going to be doing data analysis, you really don't want to compress your image. If you are going to be doing sort of presentation, putting it in a poster, a JPEG, a PNG, which are loosey compression, are good. And you can see how, if you have your original TIFF file, you compared to the JPEG, there is some data that is not really stored. So you're really losing data if you're always converting your files to PNGs or JPEGs. In terms of data management, you always want to keep up your original files. And every time you do something to the image, you should make a copy and really document what you did to the image. So you make a copy, do a different process, or export it, different process analysis. So every step of the way should be documented. And the manipulations of the images should only be done on the copy. So then if you, at some time when something goes wrong, you can always go back to your original file. In terms of backup, you always want to keep an original data file safe and unchanged. You need to always double backup your data as NHMRC guidelines. You should have 2 backups. And with the original image, modified image, all the process and the results. Very, very important, do not use our bump instrument computers to back up your files, as they do get deleted periodically. One really important point to make in here is even images are pretty, could be pretty, they are still scientific data. And if you do treat them inappropriately, that's data manipulation. And some of the things you could do could end up in really like fabrication. So it is important that image beautification, it is data manipulation. It isn't okay to make the images clearer. Well, only in a certain way. And I hope we cover a little bit of that today. So first of all, these are pretty much the guidelines for imaging. And if you really have some time and want to go through Twitter, Elizabeth Vick does a lot of, she goes through papers and finds all manipulations through images and compares the previous image or the manipulated image. So in this case, we have an example of a Western blot where, for example, a band was removed or a band was added. This is absolutely unethical. So you are changing the results of your Western blot in this case. If you do run several Western blots, they should be indicated as separate numbers. So then people can know that they are not really on the same run. Then in here we have these users modify this bit here, and they enhance some of the gold particles. So the gold particles have been enhanced. They remove this background. In this case, you guys can see this is really changing the result. And you cannot really pick and choose what you want to be highlighted. So this is unethical. In this case, you can see the original image had a fold. And that was removed. And so made it look smoother, which probably would not make a huge deal in terms of the result. But it is a specific feature modification, so that's really ethical. So if you have a fold, that is normal, like it does happen when you process tissues. and it shouldn't affect the data. You can crop it out, or unfortunately, you can re-image it. This one is quite a really impressive one. So these cells were imaged in a fluorescence microscope, and then you can see just after doing some contrast changes, how some of these cells really did not belong to the image. So if objects are copied and pasted from different images, this is definitely non-ethical, and you will get into serious problems when publishing a paper that has something like this. If you do some modification to the whole image, for example, enhancing the brightness, but the result itself is not changing, that's usually acceptable. It should still be explicitly stated on the method, so then people know the image was enhanced. But if you enhance it beyond the point where the pixels are saturated, that's really not acceptable. In this case, they have these images where they have the positive control on the sample, and they both look very similar. And then by just changing the contrast on this image, you can see that these images have not really been processed the same way. And even worse, the negative control is completely black, which tells you that probably was just a black box, not really an image. So it's nowadays really easy to catch when something unethical was done or when some misconduct happened on the papers. So you do need to be quite careful on the things you do. So the best practice is you should be taking an image that should be as similar to what you see under the microscope. If you can see it properly on the eyepiece, in most of the cases, is probably the sample like didn't work, the stain didn't work, the sample is except with the exception of the infrared, far red that you can see, or the near infrareds that you can see on the eyepiece. So if you can see it on the eyepiece, you should be able to see it on the microscope afterwards. So really in the microscopes, you can push the system to see positive signal even when there's not. So you should be really careful with what you do on the acquisition. The acquisition settings should be set on the brightest sample, and they should be kept identical between samples. If you are going to be comparing samples, you can't have laser very high for one acquisition, very low for another one. The post-processing should also be identical, and you should retain all the original files post-assessment. And of course, documenting all the process. So we have a little activity with this one. So just a little image ethics question. So you have the original image on the left and the modified image on the right. So you guys can pick which one you think, if it's ethical or non-ethical, so just true or false. And if you have any questions, please put them in the Q&A. We will address them at the end. We will try to finish around 11:30. So we are almost done with this. I'll give you last 5 seconds because that's probably pretty quick to answer. Good. So I'll show you a little bit. I have 2 more slides showing you those 2 images so we can discuss them a bit. So you can see how for the first question in our quiz, it was tissue, and it had some tissue on the outside. So the researchers really got rid of things they really didn't want to highlight. And that's really unethical because you are getting rid of some specific features. So it could change the results in this case. So in this case, the answer for this one wasn't ethically modified. So that's false. So most of you got that right. Now on the second one, in this case, it was a very simple background sort of shading correction, which is, it cleaned up the image, but it didn't affect any specific features. It should be stated on the legend, but it is acceptable to do sort of shading correction on images. So in this case, that was true. That's good. Most of you got those right. And just to sum up what we've been speaking today, in this lecture, we covered, first of all, sample preparation. We gave a little bit of advice on general sample preparation, different ways of preparing samples. Then we moved on into experimental applications. So the instrument, the techniques, we spoke about those. And then we covered a little bit about image ethics.